We see them in every grocery aisle, sweeteners that promise all the taste of sugar with 0 calories. They are marketed to us as a way to enjoy sweetness without the guilt. But human biology operates on a strict system of accounting. Our bodies process everything we consume, and there is always a physiological reaction, even when the calorie count reads 0. Recently, massive population studies noticed an alarming trend. People with high levels of erythritol, a very popular zero-calorie sweetener, circulating in their blood were showing a noticeably higher risk of suffering from heart attacks and strokes. Observing that trend is only the first step. Two things happening at the same time does not mean one caused the other. To truly prove that the chemical erythritol is actually responsible for these vascular events, scientists needed to look much closer. They had to conduct a microscopic audit to see exactly how this chemical interacts with the delicate plumbing inside the human brain. That brings us to a 2025 study published in the Journal of Applied Physiology. Researchers wanted to see exactly what happens to the cells that line our blood vessels when they are exposed to erythritol. Specifically, they looked at brain microvascular endothelial cells, which act as microscopic traffic controllers. They line the inside of the blood vessels in your brain, managing the flow of blood, oxygen, and nutrients. In the lab, researchers placed human traffic controller cells into a petri dish and bathed them in a 6 millimolar concentration of erythritol. That specific concentration is highly intentional. 6 millimolars is the exact blood level a person reaches after consuming 30 grams of erythritol, which is the typical amount found in a single standard-sized artificially sweetened drink. The researchers didn't use massive, unrealistic chemical doses. They tested a highly realistic scenario to see what a normal diet beverage actually does to these vital cells. The audit revealed 4 major disruptions. First, the cells exposed to erythritol saw a 75% increase in reactive oxygen species. This chart shows the production of reactive oxygen species. The control group, in white, is dwarfed by the massive black bar showing the erythritol group. Reactive oxygen species are essentially biological exhaust. When a cell produces too much of this exhaust, it becomes severely stressed and damaged. This next chart shows the second finding, nitric oxide production. Cells exposed to erythritol experienced a 20% drop. Nitric oxide is the essential green light molecule, telling blood vessels to relax. Losing that light keeps vessels rigid. Combining these two metrics reveals a difficult situation for the brain's plumbing. The blood vessels are simultaneously flooded with stressful cellular exhaust while entirely losing their physical ability to relax. The third finding, this chart shows a 30% surge in endothelin-1 production. If nitric oxide is the green light, endothelin-1 is the red light. It actively forces blood vessels to squeeze shut, hitting the gas on constriction. This final chart shows the reaction during a clotting event. Untreated cells successfully released a protective enzyme called TPA, but erythritol-treated cells flatlined. TPA is an emergency tow truck designed to dissolve dangerous clots. Erythritol completely disabled the safety mechanism. It is worth noting this data's pedigree. The study was funded by the American Heart Association, and researchers declared 0 financial conflicts of interest. Looking at the full biological ledger, we see highly stressed cells, severely constricted pipes, and a disabled emergency response system. These are the exact mechanical conditions required for a stroke to occur. This laboratory data connects directly back to the population trends we saw earlier. It provides a clear biological explanation for exactly how erythritol is capable of causing those elevated rates of heart attacks and strokes. However, we have to recognize the limits of this specific study. It was conducted in a petri dish, isolated from the complex balancing forces of the entire human immune system. It was also a test of acute exposure over 24 hours. A single day in a lab cannot perfectly simulate the compounding physiological effects of consuming these sweeteners every day for a decade. This single study does not mean that drinking one diet soda today will cause a stroke tomorrow, but it does prove that the mechanical threat this chemical poses to our cellular plumbing is biologically real. The scientific consensus is rapidly closing in on erythritol. The broad population risks and the microscopic cellular mechanisms are now aligning to tell the same story. There is no need for panic, but these findings do require us to make calculated choices. It is a good time to look closely at the ingredient lists of foods labeled keto-friendly or zero sugar. Whenever a food label promises a free lunch, the true cost is always hidden somewhere in the ingredients. In the strict system of biological accounting, your body will eventually have to balance the books.